Okay, picking up here with trihybrid cross. I'm filming outside, so if you hear a breeze, there's a breeze. Okay, so wind on my mic here. Trihybrid is where we have two parents that differ in only 3 traits that we're tracking. Obviously, they've got more than 3 genes, but this is from the scientist's perspective. Monohybrid, we're just following 1 trait that we want to breed for. Dihybrid, tracking 2. Trihybrid, tracking 3. When you draw this one out by hand, it looks like I have a copy. Yeah, this is a trihybrid square. It's 8 squares across, 8 squares down. It's 64 squares. Okay, this one to draw by hand usually takes me probably about 30 minutes to draw it out and keep a track of all of my pairs. You've got 6 letters in each one, three pairs. For each parent has 3 different possible alleles they're going to give, all different combinations of. We'll come back to this and talk about what that goes with, but that is a trihybrid. Okay, what trihybrid square looks like. Don't ask you to do that. You don't have any homework on that page for that. It won't be on the quiz. Monohybrid, dihybrid is all you're responsible for. We can track any number of traits. Once you get past dihybrid, you're going to probably use a computer software to project your possible offspring possibilities. A geneticist or somebody who's practiced done it a lot, you can do a dihybrid. I can do a dihybrid square in 2 minutes or less. But trihybrid, that's different. And so even back when I was in grad school, that's where they were kind of using software for that. But a dihybrid, everybody did by hand because it took just as long to pull up the program and stick your information in as it did to draw it out by hand. So I think that's still probably done by hand if somebody's wanting to figure something real fast. If you're working with stuff like that all the time, you know what to expect. You know what kind of patterns to expect in your head. It's kind of like doing math. After a while, you can just do it in your head if you're doing those types of calculations all the time. Because it is math and statistics, okay? But we don't have to stop at 3 traits. We can track any number of traits we want to track in 2 individuals that we're mating using software. When I was in grad school, well, no, I wasn't. I think I may have been an undergrad or I may have just started my master's program. I had the same group of PhD friends during both of those segments of my schooling. That there was one semester where they were upstairs in a classroom that wasn't being used. They had taken all the furniture out. They had taped butcher paper together and taped it to the floor to cover the entire floor. And they were doing a huge Punnett square up there. I do not know how many different traits they were tracking for some plant they were crossing, trying to produce it. Oops, I think I just jarred I messed up my microphone, so may have lost me for a bit. Anyway, they were tracking probability of offspring, and they would just go in, take turns whenever they had free time. They would go in and take their shoes off and go walking across the paper and take a magic marker Sharpie type thing and pair up all the possible pairs for that square. I only saw them working on it a few times because I didn't go bother them, but they worked in pairs. One would kind of stand over at the side and read off the alleles from the sides, and the person would write, because if you're out in the center of the room, you have to walk and see and write and see. And I guess you could copy down a piece of paper, but that didn't appear to be what they were doing when I observed them anyway. And I don't, like I said, I really didn't get involved in what they were doing, what plant they were working with, or what they were trying to, what they were breeding for in the end, but they were looking at possibilities. And it's possible that was actually preliminary. They were doing it before they did the breeding to see what they were going to get, to what probability they could get the actual desired outcome they wanted. How many of the offspring would be worth trying, type of thing. I don't know. But it was interesting to me that you could do that. But nowadays, like I said, we'd plug that into a computer and not deal with it. Incomplete dominance moving. This is a new section here where we have something beyond Mendelian inheritance, and you've probably already kind of seen this a little bit in the labs when we went to human genetics. That was beyond, to some extent, beyond Mendelian genetics. Obviously, we have traits that are governed by a single 2 allele pair. Okay, single pairs, let's refer to a dominant recessive trait, and that's it. But we also have a lot of traits that are covered by something else. that are not governed by just two alleles that's one's dominant, one's recessive, or has the potential. Maybe you've got two dominants, maybe you've got two recessives, but that's all there is. There's more to genetics than that. Mendel didn't know that. Okay, he didn't discover that. He was very fortunate, divine providence maybe, in the fact that he started his work with peas, which are diploid, have two sets of chromosomes, one from each parent, like us, like most animals. Plants, I keep saying all semester, they're more complex than animals. They don't always have to be, depending on the species, they may be polyploid, have more than two sets of alleles. So, or a set, one of each, you know. And Mendel actually did find that, but he didn't understand it. Okay. His first two papers, as I've said before, were on pea plants, and about, I think, somewhere around, like, I think 13 years later, it never sticks in my mind, but 10 to 15, maybe 20 years later, he actually did publish a third paper, and you don't hear much about that because that whole paper basically said, those first two papers I wrote, never mind. What happened was Mendel, being a student of the new way of doing science, which was all about following a method of some sort. Your science, your process had to be a method, a repeatable method, okay, which was a whole revolutionary idea in the 1860s, that science had to follow some type of procedure, method that could be repeated, okay. Since he was a student of that, then he wanted to make sure that he followed that. When he went to the University of Vienna, that was one of the teachers he had was a pioneer in that way of thinking and influenced him greatly. And so he did that with his pea plants. I mean, obviously he studied 7 different gene types, 7 different traits, and all of his calculations followed with all of those traits. So that supported that idea in itself. But at the same time, his idea of process, scientific processes, of method that he'd been taught, told him he had to be able to apply that to another plant in order to support his ideas, or it didn't work. It was just an exception or coincidence that he was seeing in just this one situation. So he did research on a plant called hawkweed, and nothing worked. Mathematically, nothing worked. His predictions, he could predict the outcome in the offspring. So very disappointed, he wrote a third paper saying that whatever was going on in peas was basically just coincidence. So what he had done is he had gone and he'd gotten a plant that's polyploid, and so his calculations didn't work because you have to use a different calculation. Okay. So here are some of the forms of inheritance that happen other than dominant recessive, though. Even when you're still talking about two alleles, one gene pair, we can see things other than dominant recessive as well, and that wouldn't necessarily follow his calculations either. So the first thing we're going to talk about is incomplete dominance, and this is when the heterozygote has an intermediate phenotype between that of either homozygote. And our classic textbook example is snapdragons. So you have red snapdragons, you have white snapdragons, and if you breed those two together, you get pink. And this also is one of the things that caused Mendel's first two papers to somewhat be dismissed by the few people who might have been interested in genetics at that time. Okay, from what I've read, and I've read a lot about Mendel in his work, obviously, as being a geneticist and training myself in my background training, I read that the most of the people who, is meshed with what I knew about history as well, most of the people who were interested in genetics at that time were aristocrats, and they were interested in breeding their hunting horses and their race horses and their hunting dogs. Maybe some beef cattle because they liked to eat those and had up on their manners. So there were some early breeding programs in the mid 1800s, actually back I think in the late 1700s even, possibly before that, but I just haven't read about those. Maybe they weren't documented, maybe existed but weren't documented. But anyway, it wasn't widespread in raising animals like it is today, okay, to necessarily follow pedigrees and all of that, but it was starting to be back then amongst the people who had money and leisure time to pursue that sort of thing. And what they believed, the widespread belief at that time, was that if you took an extreme of a trait one direction and the opposite extreme of that trait another direction, and you bred those two animals together, you would get an intermediate. So if you wanted a perfect happy medium, then you took the animal that showed the worst of one side of that trait and the animal that showed the worst of the other side of that trait, or maybe the best, or best and the worst, bred them together, and you get this perfect middle ground. That was the thought. Like finger paint. If you take red, you take white, and you blend together, you get pink, like a snapdragon. Except that's not what's happening. Okay, we're not blending pigments here and getting pink. Looks like it, and that's what the people at the time, if they read Mendel's paper and looked at it and said, that's not what's going on. We're getting blending. It's blending. It's dominant recessive masking stuff. That's nah. Okay. So here's an example of in plants of why they thought that happened. It looks like it. So what is going on here is you've got two dominant alleles and no recessive. And the two dominants are trying to be expressed at the same time, but neither one really gets expressed fully. So you get this intermediate phenotype that's between either homozygote, between the two. So red's trying to be expressed, white's trying to be expressed, and you wind up with pink. Okay. Codominance. Similar idea. You've got two dominant alleles again. Usually we use superscripts on this. So with blood type, I'll talk about that a little bit. We've got I super A, I super B. Shorthorn cattle, red versus white. You've got practice problem or two on that worksheet that I've put up there. Put up a picture of it. You can look at it and do those problems or print it off. And the answers are on there for you. These are kind of roan cattle. Roan means a mix. They're mottled red and white. Oftentimes they're more red on the back and white on the stomach, and then on their sides they're this white and red mix of hair color. Okay, so that's a shorthorn. Cow is known for that. Mostly the cattle are known for that. In dogs, you see it with white and black hairs being expressed. In chickens, you can see it too with white and black hairs being expressed. White and black hairs in dogs, you get your blue heeler. Okay, that kind of coat pattern look. Australian shepherd, I think. They may be more black and white, patched instead of spotted, but the blue heelers were known for that bluish cast given by the black and white hairs growing side by side. Okay, and here, the speckled hen. Okay, speckled chicken doesn't have to be hen, but that's kind of an icon of old time chickens. Is the silver lace Wyandotte or Plymouth Barred Rock, but I believe the silver lace Wyandotte here. Two different breeds have some different coloring patterns, but they're codominant. They have the black and white gene, and one gets expressed in some areas, the other one gets expressed in other areas, but they're both being expressed in the same animal. Okay. So we're just dealing with two alleles still, R and W here. Sometimes you'll see this printed as R and R prime, because early on, when I was taught this too, is R and W because red and white. But later, more modern genetics books are saying, well, it's the same trait. It's two versions of the same trait, so it shouldn't have two different letters. So let's call it R and R prime. Let's call it B and B prime. Okay, one will be, I don't know which one's which. Red is red and R prime is white, probably. Black, B would be black, and B prime would be white. Okay. But you'll see both designations on that. Okay, don't let that confuse you. It just means that they're both dominant. Okay, and they're both being expressed, but not completely, not fully, either way. So it's similar in incomplete dominance. But whereas incomplete has this blending look where you get a solid color of some sort that's halfway in between the two parents. Here, you get the two colors of the two parents both being expressed in the offspring. Okay, but what if we have more than just one gene, one allele pair that influences our phenotype, how we look? Well, that's called, or sometimes not how we look, sometimes how we function. Okay, that's called polygenic inheritance. occurs when a trait is governed by 2 or more sets of alleles, and skin color is an example of that. Okay, and I have examples of that I'll go ahead and talk about. We think there's some polygenic disorders. We have some genetic inherited disorders that we don't pretty much tell they're inherited by the fact that they run in families, but we haven't necessarily nailed down all the genetics involved in just exactly what all is being inherited. It's more than one allele pair. And maybe they've identified two, but when you start getting those multiple allele pairs, it is really hard to determine exactly how many factors are involved, how many different factors are involved in this. So this is an area that's under study, and I think it's a wide open field if you're interested in genetics, that and you're intrigued by inherited genetic disorders. It's a field where there's a lot of study yet to be done. It's really hard to define. We've mapped the human genome. We don't know what all that map means. I always tell people it's like Lewis and Clark. They mapped a lot of the West. They told where the rivers and the mountains were, but that didn't tell us what those mountains were made of. It didn't necessarily tell us where the source of the rivers were. Somewhere up in those mountains, maybe. It showed us the lay of the land, but it didn't tell us soil types, didn't tell us vegetation types, didn't tell us all that sort of stuff. We had a map for later people to go out and do that, but the map doesn't tell you everything. So just because we have the human genome mapped, it means that happened. We got that done 20 years ago or so, and we're still now trying to figure out what the map tells us. Okay, and that's going to be still going on, I think, for a long time still. So a lot of opportunity for research and discovery there. Multiple allelic traits are where we have still only 2 alleles inherited. You're going to get one from mom, one from dad, but there are more than 2 allele possibilities in the population. Okay, so with blood type, for example, there's 3 alleles out there. You can only inherit 2. Okay, and that depends on which ones your parents passed to you. And it may not even be possible for you to inherit all 3. Okay, that sounds kind of weird, but if one parent has 2 and the other parent has one of those 2 and the third one, then you've got a chance inheriting any 2 of the 3. Okay, can't inherit 3, but any 2 of the 3 you could get. But if your parents all have, my parents are both blood A, type A blood. I don't know if they were homozygous or heterozygous for that, but me and my siblings are all type A, and I'm type A, obviously, and my kids are all type A, and I'm wondering if we're not all homozygous for that. Okay, do any of us carry the recessive? Don't know. Maybe we do, and it just didn't get passed on without the dominant being inherited from the other parent. So that's what potential do we have for inheriting anything else? Obviously not a B, because a B would show, as you'll see in a little bit. But let's back up here to this polygenic inheritance and talk about that. So this is skin color, and this is our textbook example always of polygenic inheritance, seems like. And it's an intriguing one, and one I find very interesting. So I've got a video for you to watch on this. There's not a worksheet, as it says there on the note, that you need to watch and do a worksheet. No, just watch it and enjoy learning. Okay, something I'd love to do in class, but I put videos up in class and students will fall asleep and play on their phones and this and that. And here it's kind of like, well, you're on your own anyway, right? So usually I have you watch the video with a worksheet to keep you focused and awake. But maybe here you can choose your time to watch it to where that's not an issue, and you can just enjoy learning for a change instead of having to worry about, did they already say that? Did I miss that? On the worksheet, you're always worried about what's coming next, and did I miss something? And just immerse yourself in the video. Okay. But she talks about something I usually present in class, just on my own, about some research that anthropologists have done. She's one of the anthropologists talking, so I think it's more interesting if you listen to her tell it than me. So we'll go from there. I'll let you have it. What we know is that there's 3 allele pairs here, A, B, C, that contribute to skin color. I have read that there's more than that. I've read that actually they were sure there's a fourth one of D, and they projected at least 10 years ago that there was probably as many as 7 here. Now, more recently, there was stuff I read hasn't talked about that, so I don't know if that was a predominant thought at that time, and that wasn't supported in further research, or if that's just still being researched. So people are still just sticking with what we know for sure and not talking about the well we think type of thing. So this is anything beyond looking at 3 alleles here is too much anyway. So even if we had a picture of the one showing the D, the 4th allele pair, I don't know that I would want to put it up here. What this Punnett square is showing says if we took parents that had equal numbers of light alleles and equal numbers of dark alleles, the perfect heterozygote for every single allele for skin color, and mated 2 individuals with that genotype, this is the possibility of skin tones for their offsprings. Now, to have all the skin tone possible, they have to have 64 kids, and the chance that every single one of them would be different by 1 allele, probably not. You probably get a couple that have the same combination, because your probability is actually going to be greater right there across that diagonal. Okay, that we're going to have 8 kids with equal dark and light alleles, even though it's the different alleles. Okay. So, like I said, that's a lot of kids. Wow. Anyway, so you probably wouldn't see it, but there's a couple of pictures that have been supported as not being photoshopped and things like that, of twins basically who were born to parents who were heterozygous for their skin color traits, and you've got one child very pale and the other child darker. And people are like, that can be twins, and it's like, well, they can, and here's how. Okay, if one child inherited all the light alleles from one parent and the other child inherited all the dark alleles from the other parent, you've got light and dark kids. Okay, and it doesn't even have to be all. It can just be one inherited a lot of the light alleles, and the other inherited a lot of the dark alleles, and may still have a light allele, and the lighter, paler child may still have a dark allele, but it's just not showing as much because it's showing. Okay, it's not going to be as dark. The child's going to be light because it has more light alleles than dark, and that shows, you see that here. Okay, and this is how polygenic inheritance with multiple traits works for anything. This works in our skin, it works in hair shading of our hair. I've read that our eyes also, eye color, we've got a base eye color, but then the shading of that gets determined by melanin, again, the protein. This is protein distribution in our skin, pigmentation. A pigment that gives us our coloring. Okay, and so the pigments of our eyes, pigments of our hair can also be affected by other by multiple alleles. Okay. Oh, I believe this is the link to that YouTube video as a backup in case for some reason my link didn't work. I've got it posted separately in D2L below this. But if you wanted to type this in because that link got broken or removed or something, then you can type it in and look at YouTube for that. Okay, moving on to blood type. So we have 3 alleles in the population. We have I super A, I super B. These are both dominant, and then we have a recessive little I. Okay, if you have I super A, you produce the A antigen, which means you would produce antibodies against B. Okay, and I super B, you have the B antigen, and you would produce antibodies against A. Okay, and if you have the recessive little I, you don't produce an antigen on your red blood cells. So if you have a blood type, these are your possible genotypes. You either have I super A for a homozygous situation, or you are a heterozygote with I super A and a little I, which means you could pass that little I on, and somebody else could pass to your child, and your spouse could pass on little I, and therefore you would have an offspring that is, guess what? The bottom down there, little I is O. Okay. If you are a B blood type, then you have I super B, or I super B little I. Okay, again, you're masking that recessive, same scenarios above, except you're type B blood, but you could still have an O child, okay, providing what the other parent gives. If you have I super A, I super B, you are an AB blood type. You have one of each, you have both antigens, and you produce, well, you don't produce any antibodies. Okay. You can accept all blood types. You're the universal recipient in blood donation. You can take any blood type as long as it's got the proper plus and negative. That's a Rh factor. That's a totally different gene that affects the blood, not part of this gene here. This is something else. Okay, multiple factors. And so if you are little I, then you are, as I've already said, O blood type, and you produce an antigen against everything. So, I mean, you don't have an antigen, you produce an antibody against everything. So you can't accept A or B blood, you have to have O blood, but everybody can take your blood since you don't have the antigens on there. You won't trigger an antibody response. So probably if you're an O blood, especially if you're O negative, you're probably getting called by the Red Cross periodically, maybe quite often. Some students have told me that they are often called, asked to come and give blood, because the O blood type can be given to anybody, and as long as the positive and negative matches, and negative is the recessive there, and it's usually more rare. Okay. Then you can that blood could be like I said, used for everybody, so it's very, very useful. Okay. Yeah, I'm going to stop there. And wait, am I going to stop there? Yeah. I think I'll stop there, try to break this up a little bit into smaller segments. You don't have to listen to as long a segment. It's a 45-minute one like I did some time back. Okay. See you on the next one.